Hi, everyone. Thanks for joining our webinar today. It's about 11 a.m. here in Minnesota, so we're going to get started. My name is Sally Head-Dahlquist, and I'm president of Immunochemistry Technologies. I'll be presenting a short webinar today, about 20 to 30 minutes, depending on our questions. We really appreciate your taking the time to view this today. I'm going to give you a broad overview of FLIVO, which is an injectable reagent to detect cell death in vivo. And you can use it in whole live animal models to help find the right drug at the right dose at the right time. This is the second in a series on FLIVO, so I encourage you to watch the first one that we did last fall for more technical details. And of course, we'll have more webinars to come. Assisting me today is Allison Westcott, our Customer Service and Marketing Associate. Thank you, Allison. You'll notice that your phone has been muted. However, we really encourage and welcome your questions, which we'll address at the end. So just type your question into the chat dialog box on the side of your screen and select Send to Host. If you have any technical difficulties during the webinar, please send us a chat and Allison will help. She will be posting the video recording of the webinar, so watch out for an email from her in the next couple of days with a link to the recording, so you'll be able to view it again at any time and share it with your colleagues. So again, I'm Sally Head-Dahlquist. I'm president of Immunochemistry Technologies. I have a BS in genetics and cell biology and an MBA. I was one of the first employees at ICT, so I started working in the lab, and then I bought out the founders when they retired in 2016. And over those years, I've been involved in every aspect of growing the company, including the development of FLIVO, which is really exciting. So like you, we're on a mission to find a cure for diseases. I mean, here at Immunochemistry Technologies, we study cell death. And ironically, many treatments like chemotherapy have to kill your cells to cure you. And we've developed a reagent, FLIVO, that detects dead and dying cells in vivo that we can use to monitor the effectiveness of treatment in live animals so you can tell right away if the treatment is working or not. So I'll go through several examples here. So FLIVO is an enzyme inhibitor, and it measures apoptosis via caspase activity. You simply inject it IV and let it circulate for about an hour. If the treatment is working and the cells are dying, the tumor will turn green or red or another color. We currently have 4 colors available for imaging. In these pictures, these are breast cancer tumors grown on the back of a mouse. They were treated with arsenic trioxide and then monitored through a window chamber system and then injected with our green FLIVO reagent for 30 minutes to image apoptosis. Once you inject FLIVO, it will circulate throughout the body and enter the cells. If there's an active caspase inside the cell, FLIVO will bind to it and then remain inside the cell. FLIVO starts working within 15 minutes of injection, so you can tell almost immediately if the drug is working or not. Here we can easily see that the chemotherapy is working because the dying apoptotic tumor cells turn green. And we can specifically identify which tumor cells are dying because those are the green spots in the pictures. And if the treatment is not working, the cells won't turn color and you need to try a different drug ASAP. Our FLIVO reagent also comes in red fluorescence. These are SCK breast cancer tumors grown on the back of a mouse. The mice were treated for 3 hours and then imaged with SR FLIVO. And we can see that the best drug on the left is killing more tumor cells, which are bright red. And if the treatment is not working, you can try a different drug every day until you find the right one, or you can use it to help optimize the dose. In this way, FLIVO can help researchers quickly screen drug candidates. And you can use FLIVO to adjust the dose until you find the right amount with minimal side effects. These are colon cancer tumors. They're the Colo 205 tumors grown on the back of female nude mice for 27 days, and they're circled. They were treated with trail at different doses and then imaged with our red SR FLIVO using a Perkin-Elmer caliper IVUS instrument. You can see on the left the high dose of trail is killing more tumors as they fluoresce brighter with SR FLIVO. So FLIVO can help guide researchers towards finding the right kind of treatment and dose. This is a Diffie tumor xenograph, which is a colorectal cancer cell line, and it's grown on the flank of an athymic nude mouse. In this experiment, the eye, you can see, is also red, as FLIVO was administered with a retroorbital injection. It's imaged with using our near-infrared FLIVO 747 on a light core pearl small animal imager using NIR 747, which they read on the 800 channel. FLIVO can help tell you when the treatment is working best and then killing the most cells. So in this example, our NIR FLIVO at 747 was used to non-invasively monitor the efficacy and time kinetics of an experimental anti-cancer drug using to treat MIA PACA pancreatic tumor xenografts in nude mice. You can tell the drug had a maximum effect about 6 hours after chemotherapy, that's the red line, compared with the control in the blue line. And the drug kind of stopped working at 28 hours. We used this data, collected the data using a CRI Maestro imaging system at near-infrared 747. Here we excised the tumor and ran it on a flow cytometer. And we saw a 5-fold increase in detectable apoptosis and caspase activity using FLIVO. This is a breast cancer tumor, and the drug killed the most cells about 3 hours after treatment. That's the blue line. And apoptosis was noticeable even 24 hours after treatment, which is the red line. So some things we can maybe deduce from this is that 3 hours may be the time when to expect the worst side effects, as all the dead tumor cells debris may start to overload the kidneys. Or maybe we'd want to increase the dose to kill more tumor cells. Or on the other hand, it might be better to decrease the dose to mitigate side effects, or maybe wait longer between doses to allow the body to clear the dead cells and kind of recover from this treatment. In addition to monitoring different types of drugs, FLIVO can tell you if other treatments will work, like radiation. In this example, gamma radiation at 15 gray induced apoptosis via caspase activity in mouse lung cells. The dead cells are red with SR FLIVO, and the blue are healthy cells stained with DAPI. Here we're looking at the anti-tumor effects of an experimental anti-cancer vaccine, and it was working. We used NIR FLIVO, that's near-infrared at 690, on a CRI Maestro instrument, and to see that the vaccine was really working in this mouse. Well, what is FLIVO? FLIVO stands for Fluorescence in vivo. It's a biomarker. Technically, it's a labeled pan-caspase inhibitor, which is injected directly into the animal. The FLIVO probe consists of 3 key components. The label reporter tag, of which we have 4 different color options right now, which is linked to the caspase targeting peptide sequence, such as VAD, and then an FMK reactive group, which helps it get into the cell. Well, how does FLIVO work? FLIVO is an inhibitor that detects apoptosis, which is programmed cell death, by binding to different caspase enzymes. And caspases tell you which cell is dying. So caspases are the wrecking balls of your cells. So each one of your cells has its own time clock that tells it when to grow, when to divide, and when to die. And that's controlled by your DNA. Like, for example, your red blood cells live for about 30 days. And when it's time to die, caspase enzymes are activated, and they dismantle the cell from within. So when there's a problem with this process, we get cells that are growing and growing and growing out of control, and that's one reason you get cancer. And when cells die too early, you can get neurodegeneration. So the process of apoptosis, which programs cell death, is so basic and vital to life that it's seen throughout biology, even in plants. You know, you see this every fall when the leaves fall off the trees. So we can take advantage of this biochemistry of programmed cell death that normally keeps us healthy to help identify which cells are dying to fight disease and find a cure. FLIVO is really easy to use. You just reconstitute, dilute, and then inject it into the animal. And there are many ways to administer it, like through the tail vein or near the tumor or the jugular vein, or um, and then you just let it circulate about 15 minutes to an hour or more, even days. If people who have injected it for 6 hours and then imaged it, or had it in the animal for 2 days before they imaged. FLIVO is non-cytotoxic and it's cell permeant, so it gets in and out of the cells as it's pumped throughout the body. If there's an active caspase inside the cell, FLIVO will form an irreversible covalent bond with the caspase, and thereby inhibiting any additional enzymatic activity. The bound FLIVO probe will remain inside the cell as long as the cell membrane is intact. And any unbound FLIVO will leave the cell and is removed from the circulation of the animal in about an hour. In about 3 to 4 hours, it appears to be flushed through the liver and kidneys, so you can re-inject it into the same animal. So apoptotic cells that are caspase positive will retain a higher concentration of FLIVO and fluoresce brighter than non-apoptotic cells, allowing for easy imaging. And then you can also co-label the tissue and image the sample. We currently have 4 options available using different wavelengths for analysis, and we're always developing more. So we have 2 reporters using visible fluorescence, that's carboxyfluorescein in green, that's FAM, and sulforhodamine SR in red. And for optical imaging, we have 2 near-infrared reporters that dilate at 690 and 1 at 747, which may be better suited for deeper tissues. And here I've listed some of the instruments. Technology is always changing, so hopefully there'll be much more coming along. We're always looking for new things. Scientists worldwide are using our products to study many different diseases. I mean, FLIVO has been cited in over 100 scientific publications, and all of our products have been cited over 2,000 times. So you can really find a lot of information by doing literature searches on FLIVO. And there are many other applications of FLIVO that we'll look at now. Because FLIVO is a generic marker for cell death, it can be used to monitor other conditions, not just cancer, such as eye disease. FLIVO crosses the retinal barrier, so we can use it in eyes. The bright green spots on the left are dying retinal glial cells, and you can also see there's also a lot of autofluorescence of the green. FLIVO crosses the blood-brain barrier and can be used to monitor neurological conditions. In this experiment, researchers are looking at brain infections in BALB/C mice. The mice got an intranasal inoculation with the HSV-1 virus. That's the virus that causes cold sores, or a control injection. Then 7 days later, they were injected IC with our near-infrared FLIVO at 690 and imaged 4 hours later with a CareStream imager. You can see that the mouse on the left has a bad brain infection. It's bright red. And here we're looking at a cross-section of the brain. Using our green FAM FLIVO, you can see that it also crosses the blood-brain barrier, and this was used to study brain damage in diabetic rats. So the mouse on the left is an STZ diabetic rat, and then there's a control rat. These are 20-micron frozen cross-sections that were counterstained red with Nissl, so all the neurons are red, and then the apoptotic cells are green. You see those little spots of greenish-yellow fluorescence on the left. And this is a part of the brain called the periaqueductal gray, and that controls your pain sensation. And this area is damaged in diabetic rats. You have the green neurons at the left. So in addition to peripheral neuropathy in fingers and toes, diabetics may also have problems, neurological problems, just even processing those signals in their brain. Here we're looking at ischemia in rat liver, which is restricted blood flow and oxygen deprivation. These are 5-micron cryosections, which are counterstained with DAPI to label the nuclei blue. The black oval, you can kind of see at the kind of in the center, is a blood vessel, and we can see the dying hepatocytes surrounding the vessel. The green cells are apoptotic and dying, and they were stained green with FLIVO. One day, FLIVO may be used to help personalize medicine. This non-invasive monitoring and quantitation of apoptosis is possible using FLIVO. Researchers can keep trying different treatments every day until they find the right combination that works with minimal toxic side effects. FLIVO gives researchers more options and can help guide experimental protocols fast. So FLIVO really is an important tool. In summary, it is a fast and simple yet accurate way to detect caspase activity in vivo. We can detect temporal and spatial occurrence of cell death and help optimize pharmacokinetics with minimal nonspecific binding to generate better results with greater biological relevance, ultimately leading to better cures. And we're all on a mission to find a cure, and FLIVO can help. We can use it in live animals to help find the right dose at the right time, in the right place in vivo. So I encourage you to try FLIVO in your next experiment. We really have great technical support, and we'll be happy to help as you plan out your next project. And we would absolutely love to see your data, any results, so we can help continue to build a knowledge base that we can share with other researchers. It looks like we have a few questions that came in. We'll take some Q&A now. Okay, so the first question is what happens when you inject it? How does that work? Well, you inject FLIVO IV, or you can inject it near the tumor or through the jugular vein. We usually inject about 50 to 100 microliters per animal and let it circulate about 15 minutes, well, at least 15 minutes. Typically, we circulate it about 1 to 6 hours and then image or start processing the animal. Once you inject it, FLIVO is just pumped throughout the body with the natural circulation of the animal, and it goes in and out of the cells. And when it gets into the cell, if there's a caspase enzyme, the caspase binds to it and retains it within the cell. It forms an irreversible covalent bond, and the caspase is stuck inside the cell. So once bound, it's essentially too big to get out of the cell, and FLIVO will remain inside that cell as long as the cell membrane is intact. So any unbound FLIVO will leave the cell, and eventually it gets filtered out through the liver and kidneys. It seems to stay in circulation for about an hour. Some people have imaged after 6 hours or 15 hours or 22 hours or even days later. Generally, the longer the reagent circulates within the animal, the lower the background signal. But some positive signals, maybe they're apoptotic or pyroptotic, they'll still continue to degrade, and they may be lost over time as the cells continue to die. Another question, is FLIVO an antibody? No. FLIVO is not an antibody. FLIVO is it's a peptide sequence. It's a caspase inhibitor. So it is a short sequence of different peptides that the caspases preferentially bind to. And then it includes a fluorescent or near-infrared tag on it. So then the caspase binds to it, retains it within the cell, and then you can detect that signal. So it's cell permeant. It's non-cytotoxic. It gets inside the cell. There's no reaction from procaspases nor inactive forms of the enzymes. Antibodies would be difficult to get into the cells. We don't have to permealize the membranes or anything. FLIVO automatically enters the cell. It is not an antibody. Another one is, can I use it with other animals? Yes. FLIVO has been used with many different types of animals, and examine many different target organs. Mice and rats really are the most popular data that I've seen, but it's also been used in squids, bird, sparrows, and many others. If you're going to try an animal, give us a call, and we might have an example of someone who's already used that type of animal or looked at that particular organ, and we could give you some technical assistance with that. I think we'll take one more question. Can I perfuse or fix the cells? Yes. You can perfuse or fix after labeling with FLIVO. So you would inject the animal with FLIVO first, let that circulate for as long as you feel is necessary, and then start processing the animal. Some researchers have taken 2 days to process their animals, and we've seen a couple of examples in birds where they perfuse the animal, or they inject it with heparinized saline, or I think there may be a formalin they inject. Again, we have some examples of this that we can share with you if you have a specific question or need help developing a protocol. By using FLIVO, by injecting it directly into the animal first, you can get a direct result of apoptosis as it's occurring in the animal without worrying about any artifacts that may occur after you start fixing or processing the animal during sacrifice. So you just inject FLIVO, which is better than staining the tissue after the fact. It's faster and easier. And then start doing your processing, and then preserving it with fixative or paraffin. So as long as those cell membranes remain intact, you'll be able to detect the signal. If we were unable to answer your question, we'll follow up with you personally after the webinar. If you think of any more questions, please just send us an email at help@immunochemistry.com, or you can fill out the contact us form on our website. And, you can always reach us through social media on Facebook, LinkedIn, or Twitter. And of course, our website has many, many different resources to help you, including other webinars on a wide variety of topics. As a reminder, we did a deeper dive on FLIVO at our first FLIVO webinar in November, so check that out. And Allison will be sending out today's webinar as a recording within the next day or so you can watch it anytime, share it with your colleagues. Well, I think we're at about 20 minutes now, and we wanted to keep this short and sweet today. So thanks again, Allison, for managing the webinar today, and thank you, of course, to all of you who are listening. We really, really appreciate you taking your time, and please reach out to us if you have any questions or need anything else, need any other technical information. We would love to help. This is Sally Head Dahlquist from Immunochemistry Technologies. Have a great day.